Hi, this is Dr. Padma Garvey. Today, I'm going to be talking to you about research involving yoga and a plant-based diet and how these things affect aging. This research even won the Nobel Prize in Medicine in 2009. So let's find out what it was all about. Theoretically, a human cell could live forever, but because of daily wear and tear, they don't. Scientists know that they can keep some simple organisms alive indefinitely in the lab. One such simple cellular organism is called tetrahymena. Scientists are able to keep this single-celled organism going on indefinitely in the lab as long as they provide it with the right environment and the right amount of food. The cell continues to divide and the process doesn't seem to stop or die out. Dr. Elizabeth Blackburn from the University of California, San Francisco, wondered why. Now, in the human body, we have cells called stem cells, and these cells divide and multiply and serve to replace worn out or damaged cells. For instance, the red blood cells coursing through our arteries do not have the ability to divide and multiply. Every month, a woman will lose a certain amount of her red blood cells through her menstrual losses. If we didn't have the ability to replenish our red blood cells, then we would die. Luckily, in our bone marrow, there are stem cells that have the ability to divide and multiply, producing replacement red blood cells. Such a process occurs for many of the cells in our body, including even our brain to a certain extent. In order for these stem cells to divide and multiply and produce replacement cells for our organs, they need to copy their DNA code accurately. Even a small mistake in the DNA code can lead to cancer or early cell death. Now, Dr. Blackburn wondered why the organism tetrahymena was so good at living indefinitely and so good at copying its DNA perfectly each and every time. She, like many other scientists, knew that the chromosomes in organisms like tetrahymena or humans are capped on both ends with long stretches of nonsense code. These long stretches of nonsense code are called telomeres. Blackburn noticed that tetrahymena had more telomeres than most organisms. She wondered if telomeres played a role in cellular aging. A very simple way to understand DNA replication is to picture a zipper like the one on your winter jacket. Notice how the ends of the zipper have a section that holds the zipper in place, lines it up properly. If you zipped up your winter coat without those ends in place, then the zipper would fray open at the ends, it wouldn't line up properly. Telomeres are like the ends of the zipper that hold the zipper in place and let it line up properly. When a cell copies its DNA on a chromosome, an apparatus hooks onto the end of the chromosome and moves along, making a copy. If you want to make sure that all the important information is copied properly, then you want this apparatus to hook on and secure itself before it starts copying the important stuff. If this apparatus had some nonsense code to hook onto first, giving it a chance to correct its alignment and positioning before it got to the important stuff, then you are more likely going to copy the important stuff correctly. So, aging, even in humans, boils down to whether or not our telomeres are nice and long, giving our cells a chance to copy the important stuff on the chromosomes correctly. The shorter these telomeres get, the more likely mistakes in our DNA could get passed down, accelerating cancer and cellular death. Blackburn wondered what factors control how long the telomeres are. She discovered a special enzyme, another apparatus, whose sole job is to keep the telomeres nice and long. She called it telomerase. If telomerase doesn't work well, the telomeres get shorter and shorter, and our DNA gets passed down with more and more mistakes, leading to cellular aging and cancer. In 2004, Blackburn published research in the National Academy of Science showing that stress affects telomere length. Blackburn measured the telomere length on 2 groups of single mothers. One group had to take care of a disabled child, whereas the other group of single mothers cared for a child without a disability. She assumed that the chronic stress level would be higher in the group caring for the disabled child. She showed that the activity of telomerase was reduced in the higher stress group of mothers, and in turn led to their chromosomes having shorter and shorter telomeres. In 2013, Dr. Dean Ornish and Dr. Elizabeth Blackburn published a study in Lancet Oncology showing the effects of lifestyle modification on cancer patients. They evaluated 2 groups of prostate cancer patients. One group did nothing to change their life. The other group had intensive lifestyle modification, including yoga, meditation, and a plant-based diet with less than 10% fat consumption. After 3 months, they showed that the telomerase activity and the telomere length was much higher in the group that underwent the lifestyle modification. In 2015, Dr. Carlson and Dr. Blackburn published a study in the journal Cancer that looked at the effects of yoga on telomere length. This time, they had 2 groups of breast cancer patients. One group went to a support group, while the other group did yoga and meditation. They found that the telomere length was higher in the group that did yoga. There are many, many, many studies showing that telomere length is associated with a low-protein, low-fat, high-fiber diet. While all of this is preliminary, it does seem to suggest that stress reduction and eating a diet high in antioxidants, like a plant-based diet, promote the activity of telomerase and increase telomere lengths, which keeps our cells living longer and cancer-free.